Okay, so I think we'll get started then. So, good morning, afternoon, evening, night, everyone that's joining us, and welcome to the Palaeoverse Lecture Series. This is a lecture series focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Typically, these sessions will run for about 50 minutes in total, and we run this series throughout the year, which is usually on the last Thursday of each month, unless they overlap with holidays. So we'll continue next calendar year as well. If you're interested in presenting a new method or software that you've been developing or implementing, please feel free to reach out to one of the team. Before moving on, I'll give you a couple of quick introductions. I'm Harriet Drage, and with me today I have Erin Dillon, Lewis Jones, Will Gertie, and Bethany Allen. And the five of us are on the development team of Palaeoverse, an organisation aiming to bring the palaeobiology community together to share resources, reach agreed standards, and improve reproducibility in research. If you want to know more about Palaeoverse, please check out our GitHub or our website, and Lewis has just put the links to those in the chat. So that's all for me. Erin's now going to give you some tips and instructions for the webinar, and then Will's going to introduce this month's speaker. So thank you very much. Please enjoy, and over to Erin.
Awesome, thanks so much. So as Harry mentioned, the format of this seminar will consist of a talk by our speaker, followed about 10 to 15 minutes at the end that are dedicated to Q&A and discussion. So my fellow hosts and I will help field your questions and moderate this discussion. Please note that this seminar will be recorded and it will be made available on the Palaeoverse YouTube channel, as well as our website. However, we will stop the recording before the Q&A period in case you prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. So you'll see a Q&A button. You can use this to submit your questions to our speaker. And during the Q&A session, we'll either read out your question or invite you to turn on your microphone in case you'd like to ask the speaker directly. There is a raise hand button. So alternatively, you can raise your hand and we'll call on you and invite you to turn on your microphone so you can ask your question. There's a chat button. So currently you can only chat with the hosts in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. And then there's a closed captions button that you can turn on at any time to view the transcript. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when you ask your questions and interact with others in the chat. And any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. So for more information, please refer to the Palaeoverse Code of Conduct, which can be found on the website, and I believe the link is just being put in the chat right now. And with that, I will pass it over to Will to introduce today's speaker. So over to you, Will.
Thanks, Harriet and Erin, for getting us all up to speed on how the lecture series and today's lecture will work. Today, I have the great honor of introducing our speaker, Dr. Thomas Guillerme. Thomas is a NERC Independent Research Fellow at the University of Sheffield in the UK. He completed his PhD in zoology at Trinity College Dublin in 2015 and went on to do postdocs at the University of Queensland, Brisbane, and the University of Sheffield. Today, he combines classical methods such as descriptive paleontology with modern bioinformatics tools and mathematical modeling to better understand how to use morphology and evolutionary biology in paleobiology. This has led him to work on a wide variety of projects spanning the intricacies of including morphological characters in phylogenetics, taxonomic and functional diversity in a macroecological context, and disparity through time and space, about which we'll be hearing from him today. In addition to his research, he is also a science openness advocate, actively sharing his work and using platforms like GitHub and social media. He is also the creator and maintainer of two R packages, Disparity, which has been going strong since 2018, and Treats, which was published at the end of last year. Today, we're very excited to hear from him with a talk titled Top 10 of the Best Disparity Metrics to Use in Paleobiology. Number 10 might surprise you. Please join me in welcoming Dr. Guillerme to our virtual stage.
Woohoo! Thanks a lot. oops, I'm just going to share my screen. If everything works well. Nope. There we go. Everybody can see something? Yes.
Looks great.
Yes. Cool, thank you. I'm gonna just copy some links here that I'm gonna go back to. But basically just a couple of things before starting. Those that were at the at the Palace conference last week, the first 10 minutes is gonna be exactly the same. So go grab a snack, have a nap or whatever. I can wake you up in 10 minutes. But after that, we're going to do some kind of workshopy to finally figure out which are the top 10 disparity metrics. And for that, we're going to go through this vignette that I've put the link in the chat. Normally, people should see it. And if you want to follow along, there's also the row code. But don't worry if you don't want to type anything, or you can just listen to me and see what the code is doing, etc. But yeah, so top 10 disparity metrics. Not at all clickbait title. So number one. Oh, I don't think it's work. Okay. Wait, can you guys see anything? No. Okay, that's my starting joke here. Basically, I don't know the top 10 disparity metrics you should use, so that's a bad spoiler. And sorry for the clickbait, but hopefully we can figure out how to make this top 10 list together and kind of understand this kind of research I've been doing with all these people since maybe now 5-6 years, working on how to measure things in multidimensional space, both in ecology, in evolution, in paleontology, etc. So yeah, thanks a lot to these folks to having put great inputs in these projects that I'm going to talk about. So this is a question, basically I get quite often asked by colleagues, what should I use as a disparity metric to measure something? My initial response is always the classic, well, what do you think? And then I think a bit longer myself, and it's like, okay, we have to think, what are we measuring with disparity? So just to be up to date here, this is some kind of old paper I wrote that's really, really good, where they were basically looking at 3 different aspects of coiling of some shells, and then plotted the observed shells in this kind of coiling shape. So it's a 3-dimensional shape. And what you can, what kind of strikes you here is that the space is not occupied uniformly or nor randomly. So we see that like all these like shells, coiling kind of types are in a very specific corner of the trait space. And I think what we're trying to do with disparity or functional diversity, etc., is trying to understand how these spaces are occupied. Another example, quite old as well, by Mike Foote, this one is looking at this kind of space occupancy, so disparity through time. So on the x-axis, you have time in million years, and on the y-axis, you have your disparity metric, and we're going to see what we put there, but basically you have your squiggles that go up and down, and you can tell some kind of story. So this always makes me ask the question, which disparity metric do I choose to put on this y-axis to look, for example, at disparity through time? So this has been a long-lasting question. So already, again, the same author as previous paper in the 90s was thinking about which metric to use, and basically a kind of way to test whether this metric is measuring something interesting. So they started with some kind of initial distribution, so these are kind of random data points, then randomly or selectively remove these data points, and then this results into 2 distributions, where hopefully the metric we chose can distinguish between either random extinction or selective extinction if the question is about extinction. This kind of work has been done since then, has been repeated many times. These are just 4 examples. What you can clearly see here is that since 30 years, we now use colors in graphs, which is nice. I like that. And we're converging towards using blue and orange, which I think is really good. At least we agree on something, which is very nice. And then basically this kind of simulation leads to these very clear, easy to read graphs where you have some metrics, some data sets, and this should hopefully answer your question. I just have to look at it 5 seconds, and I know which disparity metric I should use. This is work we've done with colleagues, and yeah, obviously it's very clear everything. Spoiler, it's not clear. This is going to be a bit of autocritic. I really like working on these kind of projects, but since then I've been taking a step back and saying, okay, this is very nice intellectually, but this is maybe not very useful. Where do we go from there, basically? So again, to this question, what are we measuring with disparity? Going back to this disparity curve, already we can notice that this is disparity through time. And for those that know their kind of big 5 mass extinction, there's a big mass extinction happening here. So we could basically say this curve is disparity through time across a mass extinction event. So if I reshuffle these terms, we have here the mechanism. So that's the mass extinction event, a process, this is through time, and a pattern. So in a way, disparity is what, the process is how, and the mechanism is why. So just to highlight here, the mechanism is what we are usually interested as biologists, and what definitely gets us grants. If you write on your grant application, trust me, I've tried. I just want to look at different patterns. Nobody gives you funding, but if you say, I want to understand how mass extinction work, people give you funding, hopefully. So if I reiterate this question, what are we measuring with disparity? We're measuring a pattern generated by a mechanistic process. And this is going to be important, I think, to select disparity metrics. So if you can see here, again, on this example, here, just to illustrate, the process is kind of before and after the mass extinction. The mechanisms here are 2 different types of extinction, and then you can draw your pattern by measuring some kind of disparity. Okay, so knowing this mechanism, process, and pattern, how do I choose my metric? Well, I'm going to illustrate this with a very cartoonish example here. So it's very overly simplified, but hopefully we can complexify it a bit later on. So if we look at these 2 pictures, we can basically ask a question like, how is competition changing diversity through time? We can ask a lot of questions, but I've put that one for my example. So here we can easily identify the evolutionary mechanism, competition through time. Again, this is our process, and diversity will be our pattern. And so the question we want to ask here is, what pattern will capture these changes in diversity or the absence of changes to answer this question? So the first pattern could be just counting the number of vertebrates in both pictures. Or the second one, let's try 2 patterns here, is just counting the number of dinosaurs and mammals separately. So these are the numbers. I'm not gonna spend time counting them, but basically, hopefully, if you think back at what is the mechanism we're interested in here, competition, it should strike you, hopefully, that the first pattern is not going to be very useful to answer this question, whereas the second one will be more useful, hopefully. So this is the idea, and if we apply that to more disparity metrics in multiple dimensions, if we again, just an example here, what is the effect of mass extinction through time on disparity? Again, we identify mechanism, process, and pattern, and we can decide that, yeah, the pattern we're interested in is the size of the trade space, or the density, or the position of elements in the trade space. We argued in a previous paper that it's very good to use is maybe good to use these 3 elements. But I think now, since then, I feel there's many more things that we can measure. So for example, I've been doing some work with colleagues here in Sheffield, where we look at shape and orientations. So if you like spirography, you know, these ellipses everywhere, just check this paper we published last year. But basically, all these ideas here are summarized in this paper, the what, how, and why of trade-based analysis in ecology. This is focusing on ecology here, but it totally applies to any kind of other field using multidimensional trade space. So watch this space. This is now in the 2nd round of review in ecography, so it should come out hopefully soon. And it's basically summarizing what I've been saying. We're not summarizing, developing on it. Okay. But that's seriously, how do I choose my metrics? So those that were napping because they already saw this part of the talk, maybe you can wake up now a little. And now I'm gonna basically go through a practical on how basically using this kind of concept allow us to choose the right disparity metric. So like everybody, I panicked when I gave this talk title, and I just Google how to do things. Oh, this is the wrong talk. Okay, but now you know that there is, sorry, this is the wrong version of the talk. Now you know that there is Palaeoverse Lecture Series. I had another joke there, but I'm gonna skip it. But yeah, so this is it for this part of the talk. And now if people can follow troops, I'm gonna start looking at this vignette. Where basically we're gonna go at a kind of WikiHow style, like step by step, looking at how to look at disparity analysis. So for those that want to follow along, you'll have to install and load the disparity package. So you can do that while I'm kind of chatting around. But for those that just want to follow, here's the vignette. You can just, I'm going to scroll down and you can just see what's happening on screen, hopefully. For those that want to follow around, but that don't like to use this, the Shiny app that allows you also to test your metrics without using R. So it's doing things. I'm not going to go into details into how to use this tool, but you can totally use this tool. And in the vignette, it has details on how to apply the R code to this vignette. So how to select our own disparity metric. Also, I'm going to start by step 2 for this example, but I suggest people start with step 1. They can be done in any sort of order, but here I'm just gonna start with step 2 because I think it makes more sense for this practical. So the first step is to have a data set. You can of course use your own data set. So when I talk about data set here, is any kind of trait that you analyze and put into a trait space. So trait space for me is something where your rows are observations, species, elements, whatever, and your columns are dimensions. If you don't have any at hand, don't worry, there is some in the disparity package that you can use. So here's a list of all the demo data sets that you can use. They're all very nice. And you can ask different questions with it. Yeah, I'm just going to use for this demo, I'm going to use this data by Beck and Lee, published in 2014, so 10 years ago, but it's still very good for examples. If you want to use your own data set and you're familiar with the disparity package, you can use functions like custom subsets or chrono subset to make your own data. But don't worry about it now if you don't know about it. You can always look for all the functions I'm talking in the manual by just typing question mark and the function. And hopefully I scribbled a lot of info that would help you. But anyway, here we have our data set. So also part of step 2 is always something I suggest is have a look at your data set. So here we're plotting it, just using this plot function here. And here we have 2 different sets. So we have crown mammals and stem mammals and 2 dimensions. So as you can see here, this is the percentage of variance on each dimension. So we're not even at 5% with these 2 dimensions. So this is something I always like to highlight. Here we're looking at less than 5% of the data. So it's going to be very hard, at least for me, to kind of guess what's happening and guess how our metrics are going to react to these different data sets. And I think this is why, unfortunately, there is no one disparity metric that fits every data set. And hopefully through this tutorial, we'll figure out a way to find a metric that fits your data set and your question. So, okay, this is the data set. Yeah, there's basically 103 dimensions that are hidden from us. But don't worry about them. We'll tackle them using computers. So they're going to be taken into account, basically. And then back, so sorry, I'm doing things backward, but this was just to present the data set I'm using. Then the first question, or normally the first question, is identify the mechanism and the process that you want to study. So this is basically just, I find it always a bit like a kind of maybe patronizing when I teach that or anything, but I find it, at least to me, it helps a lot to write down either on a piece of paper or on your computer what exactly is your question, and then identify what is the mechanism and process, because this is going to guide you selecting your metrics. So in this case, I came up with this question: do modern mammals, so crown mammals, evolve more disparately, more disparate body shapes, than archaic ones? So looking at that, you could find some kind of evolutionary process. So the evolutionary process would be that, I don't know, ancient mammals had more or less disparity due to maybe competition or other mechanism we're not looking at specifically here, but this would be the mechanism we're looking. And the process would be like, basically, again, in this example, I think in paleontology, the process is often time. Basically, rephrasing this question is like, does evolution has an effect, as in a different outcome, on the age of the group in terms of disparity? So by putting this mechanism and process down, we can figure out which pattern to choose. So here again, I want to highlight, by the pattern, I mean which disparity metric, functional diversity metric, dissimilarity metric, space occupancy metric, name it the way you want. Basically, we want to summarize something in this trait space to tell us about the evolution of crown and stem mammals in this specific case. So then step 3, once you have these 2 steps sorted, is to look at which aspect of the trait space will help you answer your question. So here again, we want to see how disparity change in these 2 groups, the archaic and the kind of modern mammals. And in disparity here, maybe we mean the size of the trait space. So maybe the size of the trait space indicates something like a difference in diversity in different shapes. So the bigger the trait space size, the more different shapes you have. Maybe another aspect here we can be interested in is density. So this is more like to see how common maybe our different shapes in this trait space. So here I've written down a contrasting scenario where we could have a group that has a big size, that occupy a lot of space in the trait space, and also a very high density, which will be very different, hopefully, than a group that occupies a small size with a low density. So we want to be able to differentiate this, again, in this 105 dimensions on this data for this specific question. Then the next step is to make a potential list of your metrics. So there's no kind of miracle recipe here, unfortunately. It requires reading what other people have published, or taking a pen and paper and scribbling down things. So there's many papers. So the papers I was showing in the introduction slides, for example, they test different metrics, so you can get inspired what they've done here. I've been citing work I've been doing with colleagues, where you can look at the list of metrics we propose there. It cannot be exhaustive, because you can always find a different metric for your different purpose. But basically, what I suggest again is write down your list of metrics. So you have sum of ranges. In the interest of time, I'm not going to read or discuss what they measure in details, but like, I believe that these 6 different metrics can be a proxy for size of the trait space. And then I've put 4 different ones, just so there is 10, so it's not totally clickbait. We have 10 disparity metrics here, where these 4 different metrics I believe will measure something about density of the trait space. Again, you can have just 2 metrics, just 1, 20, you choose. But this is basically where, once you have done all this, where I think this maybe practical will be helpful for you. So it's, once you have all that sorted, is test which metric will work best for your data set and your question. So with that, we're going to use this function that's bit old now, maybe 3 years old. I don't remember when I published it with the package, but it's called a test.metric. So if you can guess from the name, it's testing what your metric does. And it does that by kind of transforming your trait space in a kind of predictable way, and seeing if basically this metric tracks the transformation that you have, let's say, predicted, so that basically you test whether the behavior of your metric is what you expected or not. So you do that using the shifts option. We're going to see how to do that in practice, but basically the first one is called random shift, where you basically just randomly remove data points from your trade space. This is usually your no one. You don't want your metric to capture any significant change when you just randomly change something. Another one is called the size one, where you basically remove either things from the center of your trade space or from the edges of your trade space, and hopefully this should give you a different outcome if you're, again, interested in changes of size. Another one focusing on density, where we basically remove things based on nearest neighbor distance. So things that are closer together will either be removed or preserved. Again, there's always both sides that you can look at. Another one is the evenness, which is a bit similar than the density, but it works a bit more randomly. It's basically, if you all remember that from the COVID times, is the idea of flattening the curve. So if you have kind of a normal distribution, it's kind of flattening it a bit. So it's still normal, but you have longer edges and less density in the center. So it's basically spreading your data evenly. And another one that's kind of obvious is removing things based on their position. So in this 2D example, it's removing data set from like the top corner of your trade space. In multi dimensions, removing things from the top corner of your space is always funny, because I don't know where the top corner is in 200 dimensions, but the algorithm knows, so hopefully that is good. Okay, so these are basically the different shifts we can use. And just to show how to do it in practice, you use this function test.metric. Here you have my data. That's either one of the demo data if you're following with me, or if not, that's fine, just read what's on the screen. Or it's your own data, which is much more interesting when you analyze your own data. Then you write down your metric. So I don't know if anybody here is familiar with the syntax of writing metrics in the disparity package. But basically, this is this metric is a composite metric where you have the sum, which is a function from R, and ranges, which is a function from disparity. But basically, you can write any kind of number of metrics here to summarize your trade space. And then you tell the shifts you want. So yeah, I want the random one and size one. You let it run. By default, it does only 3 replicates, which I think is good to have a gist of the metric. Maybe for publication, if you want to use that, maybe use more like 10, 20. You don't have to overdo it. I think 100 takes a lot of time and doesn't tell you more than maybe 20, etc. So, but you can increase this number. It takes longer, but then you have a better gist of what's happening. And then it creates this object, test sum ranges. That's a S3 object. So that means you can just use summary or plot to plot it or to summarize it. Yeah, summarize it. It gives you this table where here it's telling you the percentage of data it's considering. So when it's considering 10% of the data, that means 90% of the data has been removed. And here 100%, that means that no data has been removed. And basically, this is the sum of ranges if you remove 90% of the data. This is the sum of ranges for so if you go back in this one, is for the inside of the donut. So that's called increase. And the hollowness is for the outside of the donut. And then you have some tests. You can specify which tests you want. Here is just a classic LM, but I don't think it's that useful. But basically then I think plotting is more easy to read yet plots. So this is when I randomly remove data. We see an increase in sum of ranges when I randomly remove data. This is not really nice for this data set. And for looking at my question, I think this is not a good behavior, because then I will not know if my group is smaller or bigger just due to the number of data I have or to some properties of the trade space. Then you can see how it works with size here. So if we do our top 10 disparity metric, I'll put that one in the bottom again for this question and for this data set. We can try another one. Here is the product of range. So I just write prod ranges here. Yeah, just this option is just aesthetic, save steps. basically allows you to have these nice plots where you can understand a bit what's happening. The orange ones correspond to the orange things year, the blue one to year, the black to the black year. So you can see a bit what your transformation looks like. And here I think it's even worse. Most of the metrics give a score of 0, so that doesn't tell us anything. If you wonder why it gives you very weird scores, this is because of the curse of multidimensionality. Don't have time to go into details here, but if you want to read about it, I wrote some course here, so you can click on these links if you want to learn more about the fun curse of dimensionality. But then finally, I just want to show an example with, again, so then again, I'll put that at the bottom of my top 10 list. But then again, I want to show one that works well, again, for this specific case and this specific question. Yeah, the sum of variance. So again, I change the metric here, sum variances, and I just plot that. So you see here, it doesn't capture anything special when you just randomly removing data. So that's very good. It's not very sensitive to the amount of data considered. So I like that. And then here you can see, so when the size of the donut increases, so the inside of the donut, when that increases, you have a kind of gradual increase in size. It's very good. And this is a bit harder to interpret, but if your donut becomes more and more hollow, you have a decrease in the sum of variance. Okay, this is very good. Now I understand how it works. But basically, if I use this metric and I see an increase in size, I can be fairly certain that this is due to actually some changes in my trait space, not some properties of the metric, etc., not some weird thing happening in the background. So I'll put this one more on the top of my top 10 list. So hopefully this illustrates how it works. I'm not going to do it on all the 10 metrics in the interest of time, because I want to talk about another example. But as an exercise, you may be interested in doing that yourself. So you can do it later if you want. You just basically change the name of the metric here, see what it does, see if you like it, and make your own ranking of the top 10 metrics. So that's on that. And so yeah, this is making the list, but this is also a bonus I was thinking about this morning. About also how I don't think you should restrict yourself to the metrics that you can find in literature. You can also come up with your own metrics. So this stems from a discussion I had with a colleague here, Rob MacDonald. He's doing a PhD on bird color evolution or something like that. And he had a trait space and he wanted to, he came to see me and wanted to see how to measure clumpiness in the trait space. So if you think of your trait space, whoops, like that, the idea is to see, okay, here there's a little clump, here there is a little clump, another one here, etc. Kind of count the clumpiness of your trait space. Maybe our red group here has different properties than our blue one here. So I thought that was super exciting, and he basically came up with this function. There is a neater version that's more optimized now in the that we implemented in the disparity package. But basically, this is just the basics. It's just counting the number of nearest neighbors for each element and summarizing that. So this is a very nice homemade function here that you can put in your disparity pipeline. So here's just to show it if it works. And indeed, we see a difference here if we feed this to the disparity function and summarize the results. We see, okay, that the crown group has a median clumpiness of 7 and the stem group has a median clumpiness of 26. Maybe there is a change in clumpiness here between these two groups, but how do we know whether this is capturing changes in clumpiness or just some weird properties of the trait space that we didn't anticipate? This is again where testing the metric would work. So here you can do, again, you feed your new metric, counting neighbors. Here it has an additional option, radius. You can always feed any other options to your metric. And again, here we're like more interested in changes in density. And we can just plot that. Oh, I don't really like that. This is not really good, as in when there is just an increase here for random, that probably means it's capturing the number of species or specimens. And here, this is also a bit hard to read. Well, it's also not a problem. We have homemade this metric, so we know what it does. One easy way to correct for sampling bias here is just to divide by the number of species. So let's go back to the drawing board, redo Rob's function here by just dividing the number of counts by the number of elements, and then refeeding this, so counting the neighbors to metric to this function. And then we see, ta-da, that works much better. And we see the way it captures density is also a bit more interesting. Here there's a lot of dots compared to previous examples, because the metric is dimension level 2. Not going to go in the details here, but I talk about it in this vignette, so you can read about the small nuance here. But this is hopefully an example to show how you can come up with your own metrics, test them, and figure out what they're actually measuring. Again, very specifically for your own question and your own data set. And that's it for me. So if you have any question, please just ask your question. I don't know how it works from here. Oops. Maybe I stop sharing.